All right, so now we're finally ready to talk about the final arm of the immune response and the production of antibodies and humoral immunity. Now remember, I went through cell-mediated immunity first, which may have seemed like an unusual way to go, but I was following the technique of looking at the things that we understand the signaling for best. And I want you to engrave in your mind, if possible, that making antibodies against something is really sort of a default position of your immune response. As long as your body can recognize something as being foreign and of enough size and complexity in order to be recognized and bound to a receptor, we call that molecule an immunogen, and we can make antibodies against it. It doesn't matter if it's lipid, if it's polysaccharide, if it's nucleic acid, if it's protein. As long as it has sufficient shape and size and complexity and is foreign to your body, you can make an antibody against it. So I handled cell-mediated immunity, which you recall has very specific signaling that tells the body that it is an intracellular pathogen that's causing trouble. In the case of the humoral immune response, it doesn't matter whether the organism is extra or intracellular in a way, because you're not really looking at the life cycle of the organism. You're looking at the foreignness of the materials that it produces. Okay? Now, when we talk about the immune response that is necessary against things that are extracellular, clearly in those scenarios, it's true that antibody is your best response. Because if I coat something with antibodies and then activate ancillary mechanisms, I can get rid of that thing by phagocytosis and complement lysis and so forth. So antibodies basically act as tags. They will label something so that your immune response will know, I guess I need to get rid of that. And so they are dependent upon other mechanisms in order to get rid of that material in the long run. Now, if you recall when we left B lymphocytes last, they were emerging from the bone marrow and recirculating through the body. And they were naive but mature. They had never done anything before. And so if something binds to their receptor, has a shape and size and complexity in order to do that, and cross-links between them, then that cell can undergo blastogenesis in the lymph nodes and in the spleen. And blastogenesis simply refers to a burst of proliferation that the cell will go through and will culminate in production of antibodies as a result. But realize that what's going to happen during that burst of proliferation is that there will be mutational changes that will occur in the coding that gives us the idiotype of the molecule. And if you recall, the idiotype is what gives the cell molecule its specificity. So when mutational changes happen in that coding, it means that there will be minute changes in the affinity or strength of binding of the molecule for its immunogen. Now, I'm not going to change drastically. I'm not going to start out being a cell that recognized a dog and turn into a cell that recognizes bananas. I have to have a similar shape, but I can somewhat make that shape more loose or more tight, depending on the mutation and the substitution of a few amino acids. And what happens over time then is that there is natural selection between the cells who have minute differences in the shape of their idiotype, and the cell who has its receptor site occupied for the most time is the one that's receiving the most signaling to multiply more. So I end up getting natural selection for the cells who have mutated to make the best fit of receptor. Those who mutated and made a receptor that was more sloppy in its fit will not win the competition to have their receptor sites occupied. So realize that what happens over time in this process is that I develop what I call affinity maturation, an improvement of the goodness of fit of the molecules that are being made by particular cells. Now realize that if this is all that happens, I'm talking about a B cell being stimulated by an immunogen and causing this process to occur. Realize that I do not develop any memory or any longevity in my cells because of this. So what will happen is that these cells will secrete IgM or IgD of a single clonal composition. They will make it at the rate of thousands of molecules per second for a period of about 2 weeks, and then they will die. So the plasma cells, which are the factories of antibody secretion that result from this initial burst of proliferation, are very short-lived in the absence of help from T helper cells. Now, if there are no T helpers encountered, notice what's going to happen to the response. Only IgM and D will be produced. There will be no memory of that. So the advantages of the improvement of the shape of the receptor will be lost because those cells will all die. The short lifespan of the mature B cell is maybe 1 or 2 days. Plasma cells last, as we said, about 2 weeks once activated. But this is a very short-lived response, and obviously, if I don't get some help called in, all that would happen is that my B cells would make this response and then die, and more would make a response and then die, and so on and so forth. And I would never develop what we refer to as an anamnestic or memory response. Now, to look at the IgM molecule here, you'll see that it has specially evolved for the purpose of increasing the inflammatory response. It is secreted as a pentamer, as you see here, held together by what we call a joining chain. And you'll see this joining chain again when we develop the molecule that we refer to as IgA. But the fact that this molecule is a pentamer means that it is the largest of the immunoglobulin molecules you'll make, and you may have wondered why they called IgM IgM when the others, if you recall, are A and B and D and E fairly early in the alphabet. Well, the M stands for macroglobulin. It is the biggest of the immunoglobulins. And so what that means is that it's particularly good at acting like an immunological sponge. It has so many combining sites here. Notice 5 monomers put together to make a pentamer means that if a normal molecule has 2 combining sites, this one has 10. So the valence of this molecule is 10 combining sites. Now, those are all identical, so 10 identical units of a foreign substance. We refer to those as epitopes of the antigen. 10 identical of those could be bound by a single IgM pentamer, which means it's very good at cleaning up a mess early on in the process. It also is going to have most of its function inside the vascular system because it's simply too large to escape. Other immunoglobulins can function in the tissues, but IgM is simply too large. But here is the mention of the valence, which I spoke about before. And because of this very large valence, we say that this molecule has the highest avidity of any immunoglobulin. Now notice, let's not get ourselves confused here. I said a word a moment ago that indicates the strength of binding between an idiotype, the combining site, and the molecule that it's binding to. So affinity is the binding strength of an individual interaction. But when we speak about avidity, which unfortunately sounds quite similar, avidity refers to how many combining sites the molecule has. So we already said with a valence of 10, IgM has a greater capacity for binding than anything else you'll ever make again. Other than that, it is the strongest activator of complement you'll ever make. And that's because, again, going back to my molecule here, the binding areas for complement activation are found immediately behind the hinge. So if you notice, with 5 of these monomers held together, I physically have 5 complement binding regions behind each hinge. So a total molecule here would have the ability of almost 5 times the ability of any other molecule that activates complement. All right. Now, what's the purpose of activating complement? Well, remember from our discussion of innate inflammation, complement is one of those things that assists in the early attraction of cells and indeed has a function in binding to molecules in order to make them more easily opsonized. So that is an important thing to increase inflammation. But if you notice, one thing that IgM cannot do is use one of its free tails because there isn't one. And if I'm speaking about binding to CD16, which is the name for the Fc receptor, you realize there's no capacity of IgM to bind to that receptor because all of its tails are in the center of that very sterically hindered massive molecule. So it does not act as an opsonin, which would contribute to phagocytosis. Do remember, however, that it gets around that problem, as you'll see when we talk about the complement cascade. Remember, because of its activation of complement, and because complement itself has an opsonin in the cascade, the activation of IgM can contribute indirectly to opsonization through complement. It's simply that it does not act as an opsonin on its own. Now, the acquisition of help from T cells is going to open up an entire new possibility of immunoglobulin isotypes for the cell to be able to make. So as those proliferating cells are pushed outward simply by anatomy from the center of the clone in the dark center of the lymph node or spleen, they're going to receive signals either from follicular dendritic cells and T helper cells in order to survive. If the signaling from the T helper is there, then realize the two cells have to get onto the same image as far as what they're responding to. And the way that that is accomplished is very much in the same way that we spoke about with the antigen-presenting cell and the T helper cell themselves communicating in previous chapters. So what I'm saying really is that a B lymphocyte can serve as an antigen-presenting cell to enlist the aid of the T helpers as it's pushed outward from that dividing clone of cells. Now, remember, this shouldn't be intuitively obvious. B lymphocytes are not phagocytic cells. And yet it's true that when they bind something to their receptor, which are molecules of immunoglobulin, as you recall, those molecules are endocytosed, processed in an acid vesicle, and then the protein components are inserted in the membrane in the context of MHC class II. So we don't call this phagocytosis, but it is endocytosis, and because of the fact of the processing in acid, that dissolves away the invariant chain, loads the peptides in the MHC class II groove, and we're set to act as our own antigen-presenting cell. So the cells that would be attracted to this, that would be in the paracortex of the lymph node, are those mature, naive T helper cells who have been migrating around the body looking for a party. And if the T helper cell has the receptor that is complementary to the shape of that peptide in MHC class II, then very much as we saw before, we're going to have a 3-signal process that's going to cause the activation of both of these cells. The first signal is always MHC class II to T cell receptor. The second signals, some of these will look familiar. We have CD80 binding to CD28, giving a positive activation signal. We have CD40 binding to CD40 ligand. And that particular interaction is the one that stimulates the T cell secretion of cytokines that will bathe the surface of the B lymphocyte and cause it to modify its behavior further. So depending upon the secretion of the T helper cell, the B cell will be instructed as to what to do from here on in. All right? Now, that signaling between CD40 and CD40 ligand is the critical step that allows for what we call isotype switching. And the reason for that is that it causes the expression of an activation-induced cytosine deaminase. And what that involves is the production of the necessary enzyme that will allow the cell to splice certain constant domains from its germline and attach the idiotype domains to a new set of constant domains. Now, you might remember this diagram that I showed you before when we were talking about the development of cells in the bone marrow. I didn't spend a lot of time then talking about the constant domains. I was more interested in discussing the creation of what we were going to use from here on as the idiotype of the molecule. But notice that as the cell leaves the bone marrow expressing IgM and IgD by alternative RNA splicing in roughly equivalent amounts, once the T cell becomes involved and secretes cytokines onto the surface of the B cell, then one of these switch regions will be activated and the intervening DNA will be looped up and cut out. So in the case of the diagram I just drew here, notice that the cell would be instructed to change to making IgA subisotype 1. And the intervening DNA that is looped up and cut out is excised and removed so that this is only a process that can happen in a one-way street. The cell can go from making M and D to anything downstream, but notice that once I have made a terminal switch to something like IgE synthesis, there's really no mechanism for me to go backward to making another isotype. So I can only move in one direction because at each of these steps, the intervening constant domains are excised from the germline, and the cell can only change downstream. Now, all of this is under T helper control. The better trained your T helper generals are, the more direct and immediate the signaling becomes. And of course, I mentioned IgE a moment ago. There are some people who become extremely good at making IgE to their detriment, because remember, that is the molecule that's going to give me type 1 hypersensitivities. So I'm not advocating that as a good strategy for life. I'm simply saying that once you've made that choice and perhaps gotten good at it by making a lot of interleukin-4, or interleukin-13 is another one involved, then it's very difficult to go backwards. The only real hope I have in those cases of persons who have atopic allergy very severely, is to give this population of cells time to die out, which takes about 10 years of no exposure, and then try to start the process again in order to encourage the patient to make another isotype. We refer to that as desensitization. And it's a cumbersome, sometimes effective, sometimes not process, but we'll talk about that more in our hypersensitivity chapter. Now, I want to point out here that the more we study this, the more we realize that we really are in the infancy of our understanding of how the immune system works. And I don't think that they're going to be able to ask you for the precise signaling that would cause, for example, the production of IgG subcategory 1 or IgA subcategory 2. The only ones that I think can be easily tested in a way that would create a question that would actually have only one answer, which of course is their goal, is to ask about 4 and 5, because these are really quite clear-cut. All of the others, it seems to be a pinch of this, a pinch of that, and I'm making soup. And the soup on the given day is going to cause the production of this. So clearly, very complicated signaling from the T helper 2 that reflects his understanding of what would be best to instruct the B cell to do. Now, let's look at the other categories of molecules and think about why I would want to change isotypes. IgG is the most common thing for me to make after IgM and D, and I make that because of the fact that it has the largest number of effector functions, so that's a good strategic design, but it's also the most common isotype because there are 4 of those constant domain switch regions. There's gamma 1, 2, 3, and 4. So I have 4 chances, which simply says statistically that the chances are most likely, even with an untrained T helper 2 general, I'm still more likely to switch to IgG than anything else. It is an activator of complement, so again, it's going to increase inflammation. It's capable of opsonization. Notice that I have that tail that could fit into the CD16 receptor on the surface of the cell. It mediates ADCC, which I brought up to you in our chapter on cell-mediated immunity. And it's actively transported across the placenta from about 28 weeks of gestation onward. Okay? The mother actively pumps IgG into the circulation of the fetus from 28 weeks on, which is one of the things that contributes to pregnancy being an immunocompromised state. Because of its size, it's capable of distributing in and out of the vasculature. The next most likely molecule to make is IgA. Again, I have 2 subisotypes, so if we're talking statistically, I'm simply going to stumble into those more likely than I would E, which is the only remaining one that has 1. It's a dimer in secretions, and it's held together by that same joining chain that we saw with IgM. Now, this is the major component of breast milk and colostrum from the immune system, and so it plays an important role in the passive immunization of the child shortly after birth. It is not absorbed into the body, but it does coat the surfaces of the mouth, the upper GI, the upper respiratory tract, the internal openings of the eustachian tubes. And so all of that contributes passively to protecting the child from having pathogens enter via those routes. It doesn't have any known effector functions, and that may sound strange. It sounds as though I'm saying it doesn't do anything. That's really not what I'm saying. I'm just saying it doesn't activate complement, it doesn't act as an opsonin, So think of it as simply being a slimy component in the secretions of your oropharynx and digestive and urogenital tracts that makes it very difficult for things to adhere and penetrate those barriers. Now, the other thing about IgA is that as is secreted, remember it's made by plasma cells on the internal surface of your gastrointestinal respiratory urogenital tract. And as the plasma cells create the molecule, there is a receptor on the inside of the epithelium in those organs that will grab onto each molecule of IgA and physically transport it into the lumen. So that poly-IgA receptor that binds to every molecule of IgA that's made there is then retained on the surface of the molecule to protect it. We call this secretory component, and it's made by the intestinal cell. It functions in causing the molecule to have some resistance to proteolytic cleavage, because of course, it's the only molecule that has to operate within the digestive contents in this way. IgE, of course, is our bad actor, but you should remember that IgE's physiologic function is when nothing else will work. And I described this before, I believe, as the anti-Godzilla response. When you have a problem where the invader is orders of magnitude larger than anything you have to defend yourself with, then the best approach is to sort of use a flamethrower in order to make the area unpleasant for that creature to live. And IgE is really the tip of the flamethrower. It has epsilon heavy chains. There's only one switch region associated with this, and that should tell you that Mother Nature understands that this is a damaging antibody, and so I should not make it unless I've tried absolutely everything else. If I've tried IgM, if I've tried IgG, if I've tried IgA, and none of those got rid of Godzilla, then I have to get out the flamethrower. But I truly shouldn't do that unless it's Godzilla. The reason why this becomes a flamethrower is because of the receptors on the surface of mast cells and basophils. They bind to the tail of the molecule, and that causes them to degranulate and spew out all of the toxic chemicals that they have. Now, we'll speak about this more because in the United States, of course, our major manifestation of this arm of the antibody response is type 1 hypersensitivities. We don't have a lot of worm parasites in our environment, and therefore this is an immune response that becomes misplaced in some individuals. And if any of you listening to this have the problem of atopic allergy or asthma or any of those sorts of manifestations, you realize how terribly miserable this is when your immune response decides that something looks like Godzilla when it really doesn't. Now, realize that one of the other components of the humoral immune response is the complement components, which are made as acute phase reactants from the liver. Realize they're made all the time in the liver. They circulate and are one of the early sets of proteins entering into an area of inflammation. There are actually 3 ways of starting the cascade, but they're all a set of zymogenic enzymes, which simply means that they will cleave one another in sequence and result in the eventual activation of the next one in the line. So realize there are 2 things to know about these cascades, and really that's it. The first one is, how do they start? Well, the so-called lectin cascade is started by recognizing mannose binding lectin on the surface of various pathogens like fungi. And the alternative cascade is turned on probably by surface charge differences between microbial cells and our own. The final cascade, which is probably the most evolutionarily advanced, is the classical cascade, and it's the only one that is begun by antibody molecules binding to the surface of a microbe. So, once you know where they begin, realize the sequence is always 1, 4, 2, 3, 5 through 9. Okay, the only number that is out of order, of course, is 4. And as usual in science, these things were simply named in the order that they were discovered. So that order is never going to change. And if I recall that the classical cascade begins with C1, the lectin cascade begins with C4, the alternative cascade begins with C3, then notice that just because they start at different points, I'm eventually going to generate this end complex in any case. No matter what started the cascade, the goal is to make what we call the membrane attack complex. And the membrane attack complex is what punches holes in the membrane of a microbe and causes it to dissolve. So, the other final thing to know about the complement cascade is that I produce split products at each of these steps. Okay? My split products that have A's as their last unit, 3A, 4A, and 5A, are said to be my anaphylatoxins because they attract cells to various degrees and increase inflammation. 3B is my opsinin, so it enhances phagocytosis because my phagocytes have a receptor for C3b on their surface. And finally, as we said, the membrane attack complex, or components 5 through 9, is what punches holes in the membrane. So really very straightforward, not a lot to know. But we will talk about the fact in our autoimmune and our immunodeficiency chapter. We'll talk about the fact that I do have some immunodeficiency diseases that involve the regulation of complement. So I hope it makes sense to you that if these are proteins circulating in the blood, ready to go but in inactive form, easily cleaved to make the active form, it should stand to reason that I need to have some sort of control that would allow me to stop that if it were activated erroneously. And so just like your car has brakes when you need to stop, the complement cascade has a braking system at the odd number components, 1 and 3 and 5. The braking system at the C1 level is cleverly called C1 inhibitor. Gets a little bit more sticky after that. C3 inhibitor is known as decay accelerating factor, or CD55, and C5 inhibitor is also known as the membrane inhibitor of reactive lysis, and or CD59. So if the cascade gets turned on accidentally, those are the points at which your body can intervene and stop the cascade because it is innately an inflammatory process that goes forward. So once again, here is my nirvana diagram, and although I recommended to you already that you take this, print it out in larger fashion, and then laminate it, realize now we've completed the story of the design of your immune response. We have worked from inflammation and production of abscesses, and in this chapter, we went down the cascade where we either took an APC2 who came in to clean up the debris of the abscess, or we took the material directly arriving into a draining node or perhaps the spleen, and activating B lymphocytes directly by binding to their surface. So I can either have my antigen-presenting cell cleaning up the mess and carrying it to the node, or I can have the B cell acting as its own antigen-presenting cell. But regardless, the signaling on this side stimulates my mature naive T helper to become a T helper 2, who then delivers the cytokine signals to the B lymphocyte that has the complementary molecule in MHC class 2, and that signaling causes the isotype switching and memory induction and affinity maturation that I need in order to develop an anamnestic response. So notice down at the bottom of the diagram, I have outlined each of the functions of the various immunoglobulins, which you can see, whether or not they activate complement, whether or not they induce opsonization, whether or not they are functional against parasites. And just to remind you, I spoke to you last time about ADCC requiring either IgG in most cases or IgE in the case of parasites. And now, hopefully, as we've completed the story, you understand where those molecules came from so that they could be used by cells that were going to actually deliver the signal that contributed to the death. Okay? So that finishes the last of the theory of the immune response. We have only one more discussion to talk about, and that is how does the system return to baseline homeostasis. So that will be the next subject we'll talk about. But let's do some hashtag make me care here. Okay, remember what we do. I read you or describe for you the issue. You stop your video, think about it, and when you have a formulated answer, you come back and then I will tell you what I wanted you to know. So first of all, it has been established that the presence of protein in a vaccine improves its efficacy and the duration of its protective response. Can you explain this based on what you know of the development of the humoral immune response? So hit the pause button, think this through, and come back. All right, well, what I wanted you to think about here is to remember that when we speak about T helper cells, remember that they have a T cell receptor that only sees peptides. They can only recognize pieces of protein presented in the context of MHC molecules. So if I don't have peptides in something, then by definition, the only thing that is going to result is what we call the T cell independent immune response. And all I'm going to make is IgM and D. It's going to have no memory. It's going to undergo no affinity maturation. It will undergo no isotype switching. So by definition, as we move into our chapter on vaccination, realize if I have a vaccine that has no peptides in it, I am going to get a repetitive patellar reflex. It will never get stronger. It will never get better. It will never get faster. Okay? The immune response depends on T cells seeing peptide. Okay, second case. The genetic absence of CD40 ligand on the surface of T helper 2 cells is a condition which generally proves lethal by the age of 25. How would you expect a patient with such a deficiency to present? Hit pause and think this through. All right, well, I actually provided a case history for you on this subject that you can look at in your spare time, which I know you have none of. That was a joke. But regardless, If you remember what we talked about here with CD40 binding to CD40 ligand and being that final signal from the T helper 2 and the B cell, that causes the secretion of cytokines which induce isotype switching. So if I have a patient who cannot isotype switch, notice they're basically going to be in a very similar condition to the patient up above. I can't isotype switch. I can't affinity mature. I can only make IgM and IgD. I have no memory. So the reason why this becomes lethal is because these patients are stuck making IgM and IgD, and they never develop any memory to what they've done. The condition is called hyper-IgM syndrome, and most of the cases are X-linked. Again, I will refer you to the case history that you can read on your spare time. Okay, next. Opsonization of antigen-antibody immune complexes with C3b is a critical step in their removal from the blood by the spleen. So 2 questions for you. Can you explain why patients with genetic deficiencies of C3b develop immune complex disease? And can you explain why patients with immune complex disease develop vasculitis and have reduced complement levels in their blood? Pause and think. Okay, so remember, the way I remove complexes of antigen and antibody from my blood is in my spleen. The spleen every day works like a little vacuum cleaner, or maybe not such a little one. It's a large organ, but it is vacuuming these complexes out of the blood by virtue of the fact that complement components have bound to them. And so my splenic macrophages are then capable of engulfing and removing them. If I do not remove them from the blood because I have overwhelmed the capacity of the spleen, then what's going to happen is that I'm going to get those immune complexes binding in the small to medium-sized blood vessels just because of their size. And when they activate complement in that location, now they cause inflammation there. So realize, if I do not make enough C3b, I'm going to have no ability to opsonize and remove my immune complexes, so I will have more immune complexes in the blood. When I have more immune complexes in the blood, either from that or from a problem of development of those that we'll talk about in our hypersensitivity chapter, I'm going to end up damaging my small to medium-sized vasculature because my spleen is not capable of doing what it needs to do. So in both of these cases, either when I lack the ability to remove the complexes, or when I'm removing too many complexes because I have a disease that's causing me to make antigen-antibody complexes chronically, then measurement of complement levels can become a diagnostic clue. So really, these are 2 sides of the same issue. And it brings us to our discussion, of course, of why it is that it's such an important thing to have a spleen. But even in persons who have a spleen that functions normally, there are cases of chronic disease where the spleen simply becomes overwhelmed. And so remember, as we move forward, these are going to generate my type 3 hypersensitivities. And it is a corollary of those that those individuals would have decreased measurable levels of complement in their blood because the complement is being used to get rid of that overabundance of immune complexes. All right, next. Glycosyl phosphatidylinositol is a common membrane anchor on the surface of red blood cells for both CD55 and CD59. Remember, those are my brakes of the complement system at the C3 and C5 level. Can you anticipate the result of the genetic absence of that molecule, GPI? Pause your video, think this through. All right, well, hopefully the thought process you worked through is that if I have a red blood cell that has this GPI molecule on its surface, and that GPI molecule is holding my inhibitors of C3 and C5 activation, then what would happen as a result of the absence of that anchor is that my red blood cells would not have the natural ability to protect themselves from complement components in the blood. Well, complement is always in the blood. So if I do not have that way of protecting myself, it should stand to reason I am going to have abnormal levels of hemolysis. The disease, of course, is called paroxysmal nocturnal hemoglobinuria. Okay, paroxysmal nocturnal hemoglobinuria, PNH, results from the genetic deficiency of that anchor molecule. And the reason why I see nocturnal hemoglobinuria, in other words, obvious red in the urine at first micturition in the morning, is because probably of respiratory acidosis when we breathe at night more shallowly. That activates complement and causes the increased level of hemolysis. It's happening at other times as well, but probably the concentration of the urine in the morning makes it more apparent then. Okay? Finally, our fifth question here. Mycobacterium leprae is an obligate intracellular organism. It can't be grown outside of cells, but inside the body, it lives in my mononuclears. There are two polar forms of the disease, leprosy. The more serious one, the lepromatous form, results when the alternative APC2-TH2 axis of the immune response from this chapter is activated. Can you think through, using nirvana, why this would be so? Pause your video, think this through, get out your nirvana diagram, and follow the pathway. All right, well, that may have taken a little bit of thought, but if I've got an organism living inside my mononuclear phagocytes, I hope that you got to the point where you realized that the best pathway to cure that pathogen is to turn on an APC1 TH1 response, and then make the necessary interferon gamma that would cause the APC1 to become a better killer of what is inside it. This was that special loop on the left-hand side of the nirvana diagram that was reserved for those pathogens that live inside your mononuclears. Why hunt them down and kill them if you can make them do their job better? So notice that although this would be the pathway I want to use to take care of that sort of pathogen, the person who made the lepromatous form of the disease took the other pathway. They took the antibody pathway, and antibodies don't do you any good for things that are hiding inside a mononuclear phagocyte. Realize additionally that if I turn on the APC2 TH2 response, as we did in this chapter, and I eventually cause the increased production of antibody, remember that the activation of that side of the immune response is actually going to inhibit the development of the cell-mediated side. The two sides mutually inhibit one another simply to concentrate one's energy immunologically on the pathway that will do you the most good. So the more I have this pathway turned on, the more I turn this pathway off. And so it generally happens to be true in patients with leprosy that after that initial decision of whether to make an APC1 or APC2, their forms of disease, tuberculoid on this side versus lepromatous, those forms become more and more pronounced over time because of the reciprocal inhibition of those two pathways. It's not an absolute, but it does make things tend to get better or worse depending on which side you went. So that finishes the last of our strategic discussion about the immune response. When we meet again, we'll discuss memory and the reactivation of the immune response and the development of the return to the homeostatic level of the immune response when things are not necessary anymore. So I will leave you with this list of things that you should ask yourself to make sure that you can indeed teach a colleague how this works. See one, do one, teach one. It's the same old medical principle. So I want you to think about all of these, make certain that you feel comfortable with what we've done, and then we'll be ready to move on and use the practical applications of what we've learned.